Wow, that's loud. I am here to introduce Sylvia. Will is also in spirit here to introduce her, but unfortunately he also really wanted to get coffee, so, you know. But we actually have some, oh wait, sorry. This is a technical challenge, so bear with it. Hopefully you'll be able to hear Will's introduction as well.
One of the most endearing things about Sylvia is how environmentally conscious she is. For example, anyone who's had lunch with her knows that she literally carries around a titanium spork in her backpack at all times, which she uses to eat from the mason jars she packs her lunch in.
So, yeah, Sylvia does really like her spork, and as Will and I were investigating this a little further, we came across this Amazon review that she seemed to have written earlier this fall. So another thing that we all know about Sylvia is that in addition to loving her spork, she just generally loves being environmentally conscious. And that goes to most parts of her life, including lab. So here we have a lovely picture of Sylvia in lab taking advantage of her mason jars, which she uses for everything. And also the fact that CSAs always give you too much food, and it's nice that she then carries that on to being resourceful in lab. Here we have another word from Will. This is great.
Sylvia clearly cares about the environment, but what she's really passionate about is her undergraduate institution, Caltech. As we were rooting around in her desk drawers in preparation for this introduction, we found entire notebooks filled with doodles like this one.
So another part of her Caltech experience that Sylvia loves to talk about is the Senior Ditch Day, in which it's a sort of a mysterious event that involves a lot of pranking and whatnot. We don't entirely understand it. One of them that she has talked about involves literal digging of ditches, which is a cute play on the word. But when we looked into it a little bit further, we found some other disturbing pictures that seemed to imply it was maybe not so innocent, and Sylvia, we'd like to talk to you about this later. Then our last word from Will here.
Sylvia clearly cares about the environment.
There we go.
Sylvia came away from Caltech with tons of amazing stories. She also came away with dozens of t-shirts, which at this point constitute most of her wardrobe. For example, this t-shirt from one Senior Ditch Day is a mashup of the classic Nintendo game Legend of Zelda and somewhat inexplicably My Little Pony. In fact, My Little Pony seems to be a recurring theme across her Caltech shirts. Like the Ditch Day shirt from the following year, which was a mashup of My Little Pony and the classic Children's Adventure game Grand Theft Auto. Darcy and I have tried many times to get Sylvia to tell us about that Ditch Day, but she says the less we know, the better. The following year, they went with a My Little Pony theme that was a bit more whimsical.
So to conclude, Sylvia, good luck, and also we found your spork earlier yesterday, so you can
All right, so recently I've had a lot of things on my mind. How do I give a good journal club talk? How do I figure out what lab I want to join? But at the end of the day, I'd like to think that I'm a relatively simple person. All that I really want is a roof over my head, some nice warm food, and the company of friends. And when you think about it, this is what most organisms want: a reliable habitat and a reliable food source. But for some, it's a struggle. And the struggle is especially real if you're a pathogen. Specifically, when you're a pathogen and the host inevitably finds you and inevitably tries and gets rid of you. This is pretty similar to how our immune system works. Our immune system needs to be able to recognize foreigners and call in special units like the police in order to get rid of any pathogens. So what else do we think of as being key characteristics of the immune system? Like I said, we think of it as needing to be able to perform self versus non-self recognition. We think of it as having a mobile specialized defender, like the police. We think of it as having things like antibodies and and memory. But if we think a little bit more carefully, we realize that all of these characteristics are really only things that are found in the vertebrate immune system. And other organisms, like plants, lack many of these characteristics. So if you're a plant and you lack something like a mobile defender cell, how are you able to defend against pathogens? How do you fight against home invaders if you don't have a police? In the case of plants, every cell needs to individually be able to detect and fight against pathogens. So if you return to our analogy here, it's like if instead of calling the police, our stick figure got rid of the problem himself. So how exactly are plants able to do this? So here we have a plant cell. And for all of you non-plant people in the room, which is everyone, the ableplast is the region between cell membranes where material gets transferred. It includes things like the cell wall, which is why the cell wall is not explicitly drawn here. So pointing into the ableplast, we have these receptors such that when our bacterial pathogen comes along, the receptor binds and recognizes the flagellin proteins, which in turn causes a downstream signaling cascade leading to an innate immune response. This can lead to things like increase of reactive oxygen species and strengthening of the cell wall. Now, given that plants have devised a mechanism to specifically recognize bacterial pathogens that are in the ableplast, why do our bacteria even bother invading the ableplast to begin with? Why not find some other region where they can more easily evade detection? And the reason for this is that the ableplast is a pretty sweet place to live, literally. So, like I said, the ableplast is used to transfer materials between cells, and this includes sugar. So we see here proteins which transport sugar from inside to outside of the cell, and other proteins which transport sugars from outside to inside. These sugar importer proteins are called originally sugar transporter proteins, or STP. So when a bacterial pathogen comes along, it can feast on all of these sugars and proliferate. What's more, it can increase the amount of sugar, making the ableplast an even sweeter place to live, and it can do this by upregulating the transcription of these sugar exporters, thus increasing the amount of sugar in the ableplast and allowing it to proliferate. So given this, people have wondered for a long time whether or not our plants could fight back against these bacterial pathogens, whether or not they can fight fire with fire by changing the activity of these sugar importers, thus sequestering sugar away from our bacterial pathogens in the ableplast and reducing its ability to proliferate. And so some pretty good evidence, although circumstantial, came about in 2014. What a group did is they took fungal pathogens and inoculated it onto some plant leaves. These specific pathogens are necrotrophic, meaning that they kill plant cells. So if we wait 3 days, we see some lesions form, which are the result of dead tissue. What they did was they repeated these experiments with 3 different strains of plants. One that has normal sugar importer expression, one that has artificially increased expression, and one that has artificially decreased expression. And given our hypothesis, we would expect that the plants with increased expression are less susceptible to our fungal pathogen, and the ones with decreased expression are more susceptible to our pathogen. And by characterizing the size of these lesions, this is indeed what they saw. So this provides some pretty circumstantial evidence that, in fact, plants can modulate the activity of our sugar importers during pathogen attacks. But just because something in biology can do something doesn't necessarily mean that it does. So that brings us to our paper here today. Regulation of sugar transporter activity for antibacterial defense in Arabidopsis. This paper was published in Science in November of last year, and it was published out of several groups in Japan. And so the question that these people had was pretty simple. Are plants able to actively regulate these sugar importers for antibacterial defense? And as you can probably guess by the title, the answer is yes. But in order to do this, they first needed a way to actually be able to measure sugar transporter activity. So to do this, they quantified the amount of monosaccharide uptake in Arabidopsis seedlings. What they did was they took seedlings that either had or had not been exposed to our bacterial pathogen. They placed them in a beaker full of radioactively labeled glucose. After 2 hours, they removed the seedlings, washed them and whatnot, and placed them in this special solvent that fluoresces when exposed to the radiation caused from radioactive decay. And so by being able to quantify this fluorescence, you can then get an idea of how much sugar our seedlings uptook during this time period. As a reminder, we have two proteins of interest. We have our sugar importer proteins, STP, and we have FLS2, which recognizes our bacterial pathogens. And specifically what it recognizes is FLG22, which is a conserved 22 amino acid sequence in the flagellin proteins. And the reason why I bring this up is because this specific protein is what they use to induce an immune response. So what they find is that indeed we do see that our plants are able to increase their amount of sugar uptake when they perceive that they're under attack. If we knock out this FLS2 protein, we no longer see a significant increase in glucose uptake. And this isn't terribly surprising because we've just knocked out the protein that senses what we've exposed them to. But what they also found is that when they knocked out two sugar transport proteins in this family, STB1 and STB13, they also saw no difference between them during the immune response. And STP1 and STP13 are just two specific proteins, and the reason why they use them is because they're some of the most important ones for monosaccharide uptake. So from this, the authors conclude that sugar transport is regulated during bacterial infection. Now, from this, they don't really have a good clue of how it happens, just that it is. But from these data, given that the FLS2 and this STP double knockout have similar phenotypes, they come up with a hypothesis, which is that FLS2 regulates our sugar importers. Now again, they're not really sure how, but given that these are both membrane proteins, they postulate that perhaps there is some sort of physical interaction between these two proteins. So in order to do that, in order to see whether or not this is actually the case, they perform a co-IP, or co-immunoprecipitation. I'm sure that many of you know what this is, so this will be relatively quick. What we do is we take our protein of interest and we attach some sort of tag. We then wash our cell lysate over a surface that contains an antibody that binds specifically to this tag. We can then remove anything that's not this protein or anything that complexes with this protein, and then later we can purify this protein complex and analyze it using, in this case, a Western blot. And what they find is that, indeed, FLS2 forms a complex with our sugar importer, and here we have GFP as a control. Now, given this, what exactly might FLS2 be doing to change the activity of our sugar importer? So you can imagine there's sort of two very general mechanisms. One is where upon binding to our sugar importer, our protein induces some sort of conformational change that in turn changes its activity. The other is that upon binding, it signals for some sort of post-translational modification, which either innately changes its activity or signals for it to form some other complex. So you can't really tell so luckily, our researchers had a way to be able to tell the difference between these two possibilities, which is they realized that STP also forms a complex with BAC1. Now, BAC1 is a kinase, and upon binding to flagellin, FLS2 and BAC1 form a complex that results in this downstream signaling cascade that I was talking about earlier during the innate immune response. So given that we know that STP13 forms a complex with the kinase, they wanted to know whether or not our sugar importer could get phosphorylated. To test this, they used an in vitro assay. What they did was they took their protein of interest, mixed it together with a kinase, so STP13 and BAC1 in this case. Together, they mixed in radioactively labeled ATP. If our protein can in fact get phosphorylated, we see that we add our protein now has a radioactive label, and when we purify this sample using an STS page, we now have a band which is radioactive, which you can then detect and infer phosphorylation. So if we look at the STP13 protein, we see that reasonably there are two regions where we could expect for it to be phosphorylated. Either in this middle loop region here, or in the C-terminal tail. Now, because this is a transmembrane protein, what they actually do is they take fusion, they take the middle loop segment or the C-terminal tail and they fuse it with some sort of cytosolic protein and see whether or not these fragments can get phosphorylated. What they see is that the middle loop cannot get phosphorylated while the C-terminal tail does. So after this, they wanted to know exactly which residues in our C-terminal tail get phosphorylated. Based on the sequence, we see that there are 5 amino acids which can get phosphorylated, and when we mutate every single one of them to an alanine, we no longer see any phosphorylation, which is not terribly surprising. When we mutate every single one of them, except for this threonine 485, we now do see phosphorylation. And when we mutate only this threonine, we no longer see phosphorylation. So from this, our authors conclude that this threonine 485 is phosphorylated by BAC1. All right, so we have our protein, and we think it gets phosphorylated. But does it actually change its activity in any way? To measure this, they return to their monosaccharide uptake assays, but this time in a simpler organism, specifically yeast. What they do is they take a culture of yeast, performing a similar assay as before, they add radioactively labeled glucose. They then filter this culture, place the filter paper in this solvent that fluoresces when exposed to the radiation caused by radioactive decay. In this experiment, they have they utilize 2 different mutants. One where they mutate this threonine to an aspartic acid, and this acts approximately like a phosphorylation, so it's a phosphomimic, and one where they mutate this threonine to an alanine. So now this protein can no longer get phosphorylated. They perform these experiments in a strain of yeast that can't take up hexose, so we see that when we introduce our sugar transporter, we see a significant increase in glucose uptake. When we instead introduce our mutant, our phosphomimic mutant, we see a significant increase in the amount of glucose uptake. And when we now instead introduce our mutant that can't get phosphorylated, we see approximately wild-type-like behavior. So given this, we can conclude that phosphorylation probably significantly increases our glucose uptake, and by mutating this residue to an alanine, we haven't irreparably broken our protein or anything, which allows us to use it in later studies. But even though we think phosphorylation might be doing anything, at the end of the day, what we really want to know is whether or not this has an effect on the antibacterial defense of the plant. So in order to do this, we need to be able to somehow measure the success rate of our bacteria. So we need to be able to quantify the bacteria that have grown. To do this, what they do is they spray their leaf with a solution full of bacteria. After several days, they cut out a small leaf disc and put it in a buffer. They perform several serodilutions until it's dilute enough that they can plate it onto an agar plate and count the colonies of the bacteria, and based on the number of bacteria, they can infer how many leaf bacteria were on the leaf to begin with. What they find is that when they perform a double knockout of both STP1 and STP13, we see that the plant is significantly more susceptible to leaf bacteria. So there's significantly more bacteria. When we have this double knockout and we rescue it with STP13, we see an approximate wild type-like behavior, and the plant is no longer more susceptible. When we try and rescue it, though, with this alanine mutant that can't get phosphorylated, we see that, again, a significant increase in leaf bacteria compared to wild type. And in fact, it's just done just as poorly as our double knockout to begin with. So from this, the authors conclude that phosphorylation of STP13 decreases bacterial proliferation. Now, this brings us to the working model of our paper. We have our plant cell, we have our flagellin receptor, and our sugar importer. When a bacterial pathogen comes along, it binds to this receptor, which in turn indirectly phosphorylates our sugar importer. This changes its activity so that it increases the amount of sugar that it imports into the cell, and thus it sequesters sugar away from the pathogen. But if you remember from our beginning of my talk, this bacterial pathogen can fight back. It can increase the expression of our sugar exporters, thus increasing the amount of sugar in the apoplast. And so what we see is sort of a fight between bacterial pathogen and host, where they're fighting over a very basic resource, in this case, sugar. This model is made even more convincing when we look at the conservation of this threonine that gets phosphorylated in our sugar importer. We see that amongst STP13 homologs, it's conserved amongst all these different species of plants that have diverged around 100 million years ago. Whereas amongst other members of our sugar importer family, this residue isn't conserved at all. So this suggests that perhaps this is a mechanism, a conserved mechanism to fight against bacteria. Now, of course, with any paper, I have my criticisms. The first being that although they perform phospholipid mutants in yeast, they never perform these mutants. They never have these mutants in Arabidopsis, and this seems like a very obvious next step. So I'm not entirely sure why they never did this experiment. The second is that although they show some circumstantial evidence that our protein here gets phosphorylated in vitro, it's never really conclusively shown that it gets phosphorylated in vivo. And thirdly, all of the experiments that they do are, many of the experiments that they do have to do with glucose uptake in entire seedlings. But that's not really what we're interested in. What we're interested in is the amount of sugar that's in the apoplast, and specifically how the flux of sugar in and out of the apoplast changes during bacterial infection. They do, in fact, have one figure where they show the apoplastic sugar levels, and they sort of shove it away in the supplemental information. And I'm not that surprised. So what we see is that when our plants think they're under attack, there's not actually any increase in the relative glucose or decrease in the relative glucose levels in the apoplast. If we knock out our sugar importers, we see a significant increase in the glucose levels in the apoplast. Now, I'm not entirely sure why it is that the apoplastic sugar levels are increasing in the first place, given that this is just flagellin protein and not bacteria. It's possible this has to do with sort of an organismal reshuffling of sugar, but it's not entirely made clear in the paper why we see this happen. And when we knock out FLS2, we see that there's no significant increase, which again isn't very surprising given that now our plants can no longer detect this protein. With that, I'd like to thank my coaches, Vlad and Susan, as well as Yichun and Jack, and I'd be happy to take any questions. Annie?
Do you have any idea about like what kind of structural changes might take place in the flagellar receptor in order to allow it to interact with the sugar importer? And also, related question, does it need BAC1 in order
So they never actually do the experiments to see whether or not they interact like without BAC1, so that's not known. As for structural information, I'm not entirely sure, and also they do in fact show that the two proteins can bind even when there's no flagellin. So there's not really a structural change that's going on to allow it to bind. I imagine there's some sort of structural change that happens upon flagellin binding that allows it to this receptor to also bind to BAC1 to then allow it to phosphorylate. Yeah?
So I was wondering, do you know if plants have commensal microbes that would protect them against invasion? Because one thing we see, like for example, in the human microbiome, there's this thought that when you're sick, you're not eating food, but then you're putting sugars on your proteins in your gut to sort of support commensals and prevent invasion from pathogens. So I'm wondering if those data there with increased sugar availability on the leaves, sort of, do you know if that could be a potential idea?
That's certainly a potential. Unfortunately, most of like the research that I came across seemed more focused on host pathogen interactions. So yeah, that's not really, unfortunately, that's not really an area that's being very heavily studied right now. Sean?
What about other sugars, like, for example, sucrose, which is the main transport sugar?
In terms of...
Like, when they say glucose, is it just, like, glucose, or does that include, like, they, like, enzymatically cleaved other sugars to your glucose? Or do they talk about other sugars?
Throughout the paper or in a specific experiment?
Just generally throughout the paper.
So
more than just
yeah. So when they do their uptake assays, they do them on both glucose and fructose, which is what these transporters transport, these STPs. In terms of this apoplastic sugar levels, I mean, they're measuring specifically glucose, and it seems like glucose and fructose are the specific sugars that they talk about throughout the entire paper. Yeah?
So it seems like the sugar effect could also be affecting intracellular processes in the plant leaf.
Mm-hmm.
And in the alanine mutation, did they look at all at the downstream events? You know, maybe if they don't have enough of an energy source, they can't really build up the cell wall the way they need to, or they can't do all these other things that they're supposed to do.
Yeah.
Did they look at any of those things?
I mean, that would be a great experiment, but no.
No.